Good morning. Hello, everyone, and welcome to the closing day of our annual meeting here at the Neurobiology Institute. I am Dr. Aleph Prieto, and today I have the honor to be the host of this session, where the keynote speaker is Dr. Matt Blurton-Jones. Matt is a professor in the Department of Neurobiology and Behavior at the University of California, Irvine, and he's also director of the UCI AGRC iPS Cell Core. He's also director of the UCI Stem Cell CRISPR Core. And I want to tell you that Matt holds a very Californian scientific career, as he got his PhD in neurosciences at UC San Diego, and thereafter he did postdoctoral studies at UC Irvine in the labs of Dr. Carl Kattman and Dr. Frank Laferla. His research is focused on understanding the molecular mechanisms that drive the development of Alzheimer's disease. Notably, his early studies were among the first to show that neural stem cells can improve cognitive and motor function in transgenic models by elevating levels of BDNF and enhancing plasticity. Overall, the work in the lab of Matt uses highly sophisticated models. For example, his group is developed one of the leading approaches to differentiate patient-derived iPS cells into microglia and generated chimeric models to study human microglia function in vivo. Importantly, Dr. Blurton-Jones has been extremely successful in securing funding. Since the beginning, his lab was awarded by multiple agencies such as the NIH in the US and also from the Alzheimer Association. I met Matt when he started his lab at UCI, and I must tell you that the work at Matt's lab rapidly fulfills all the expectation in the university. So I was very glad for him and for the opportunity of collaborating with his lab. Thanks, Matt, for accepting our invitation and for giving this talk.
Thank you so much, Aleph. That's a very generous and kind introduction. It's my sincere pleasure to speak with everyone today. I, of course, wish I could be there in person, maybe another time, but for now, we'll do our best via Zoom. So let me just go ahead and share my screen and bring my slides up. Are you guys seeing this?
Yes.
Okay, great. And then I just want to also see if I can add a pointer that might come in handy along the way. Okay, so as I said, it's a pleasure to speak with you today, and I look forward to the discussion at the end. Hopefully meeting some of you at least via Zoom. So as Aleph mentioned, we've been working on microglia for the last almost 7 or 8 years. And specifically, we've been using stem cell models to try and understand the role of microglia in Alzheimer's disease. So first, just a very brief introduction of Alzheimer's disease. I'm sure you're all very familiar with Alzheimer's disease because it's the most common cause of age-related dementia, and it gradually destroys a person's memory and ability to learn and reason and carry out daily activities. And of course, by the time someone passes away from advanced Alzheimer's, there's significant atrophy of the brain. You can see here on a coronal section on the left side is a healthy age brain. On the right side is a brain from someone who passed away from advanced Alzheimer's. And you can see this widespread atrophy, especially within the cortical regions, a big increase in the lateral ventricle, and this big hole here that should look like this, which is, of course, the hippocampus. And we know that the hippocampus is critically involved in learning and memory. And so it comes as no surprise that those are some of the key symptoms of Alzheimer's disease are impairments in those functions. We know also that the prevalence of Alzheimer's is increasing, and this is largely due to demographics. So as our populations begin to age, of course, is the biggest risk factor for Alzheimer's disease. And worldwide, there is an aging population. In the US, they call them baby boomers, but there's a similar phenomenon around the world, such that the number or the proportion of our population that are reaching the ages of 75 or 80 or 85 is continuing to increase and projected to further increase by 2050. And so if we can't come up with a good, effective treatment to slow the progression of Alzheimer's disease, this is going to continue to become a really pressing healthcare challenge across the world. So the field has known quite a lot about Alzheimer's neuropathology, beginning over 100 years ago when Alois Alzheimer's, a German neurologist, first described the pathology that he was seeing in these patients that had these cognitive symptoms. And so he described two key hallmark pathologies. One is amyloid plaques, which are shown here. These are these brown deposits that accumulate extracellularly, and they're made up of a small protein called beta amyloid that seems to aggregate and fibrilize and become insoluble, and our brains have a hard time clearing this deposit. And that seems to occur very early in the disease. In fact, often 15 to 20 years before symptoms start to appear is when this pathology starts to accumulate. Alzheimer also described a second pathology that occurs within the neurons, and that's neurofibrillary tangles. Here we have an accumulation of a different protein, a protein called tau, that becomes hyperphosphorylated and insoluble and impairs neuronal function, axonal transport, eventually leading to neuronal death. This is all accompanied by various forms of inflammation. So for instance, you have both microglia and astrocytes becoming reactive in Alzheimer's patients. And this image here is showing an example of some microglia in red that are surrounding an amyloid plaque in blue. And you may just be able to make out, there's a few little blue dots in some of these microglia. One of the things that microglia try to do is to clear this amyloid via phagocytosis. But it seems that in our aged brains, there are some real challenges with the ability for microglia to clear this insoluble amyloid. And so they're not very effective at that. All of this together, all of these pathologies together lead to neuronal and synaptic loss. And that, of course, is what really underlies the cognitive impairment. The best correlate to dementia and Alzheimer's disease is the amount of synapses that are lost. And there's a good amount of evidence that each of these pathologies can individually cause synaptic loss, but also in combination can exacerbate each other. So we know that Alzheimer's disease, even what we refer to as late onset Alzheimer's disease, which is the great majority of cases, about 98% of Alzheimer's cases, are still highly genetic, but they are polygenic. So they are affected, the risk of developing this disease is influenced by multiple genes interacting. And one of the clearest examples of how we know that they are genetic is from looking at twin studies. So you can see here, these are a number of different genetic diseases, and Alzheimer's disease is quite high in terms of the concordance. So if one identical twin has Alzheimer's, you have a little over a 60% chance that the other identical twin will have Alzheimer's disease. That suggests highly genetic, although clearly there are also behavioral lifestyle effects that are environmental effects that are influencing whether someone gets Alzheimer's disease or not. So what are these genes? Well, that's where some real remarkable progress has been made over the last decade. And that's through genome-wide association studies that have looked at tens of thousands of patients in order to find these genes that on their own can have fairly subtle impact on disease risk, but when taken as a whole, when combined, can lead to a substantially increased risk of Alzheimer's disease. And so we now know, I think, over 50 or 60 genes and loci that have been linked to Alzheimer's risk, some of which seem to increase risk, others might be protective. And this is a subset of them shown here, some of the key ones that have been validated, and looking simply at what cells in the brain express the highest levels of the mRNA for these genes. And so this is taken from a database in which mRNA was isolated from different human cells, so astrocytes, endothelial cells, microglia, neurons, and oligodendrocytes. And the heat map here is simply showing what is the relative level of mRNA for each of these Alzheimer's risk genes. And you can quite clearly see that many of these genes, arguably 3/4 of them, are most predominantly expressed in microglia. And so that's sort of renewed the focus on microglia in the Alzheimer's field, going from thinking that maybe microglia is a late stage response to the pathology to actually understanding that the genetics is suggesting microglia are much more importantly involved in this disease than we had previously thought. So what are microglia? Well, they do a number of very important functions in the brain. They're, of course, immune cell. They are the resident innate immune cell of the brain. And so they're important for immune surveillance. If one's unfortunate enough to get a bacterial or viral infection in the central nervous system, the microglia are going to be critical for that defense and have been shown to be very important for the protection against those kind of insults. But most of the time, fortunately, we have a blood-brain barrier that prevents those kind of infections. And so microglia actually predominantly play a very important role in just maintaining normal neuronal homeostasis. They can provide growth factors to neurons that help regulate neurons. They're also important in turnover of other components of the brain. So for example, myelin undergoes turnover, and microglia are critical for the clearance of older myelin so that new myelin can be laid down. They, of course, can respond to injuries, such as traumatic brain injuries. And they can clear unwanted extracellular aggregates. And I showed you earlier how they try to do that in the case of beta amyloid. And perhaps in some patients, this works quite well, but perhaps in others, there's an impairment, and that might be contributing to Alzheimer's disease. And then lastly, there's some very nice work, mostly coming out of Beth Stevens' lab at Harvard, showing that microglia also play an important role in regulating synaptic plasticity. So they can prune unwanted spines to allow new memories to be laid down and new plasticity to take place. So in terms of these Alzheimer's disease risk genes that I mentioned, one of the things one can, of course, do is turn to mouse models where you manipulate that gene and ask, what does it do to microglial function? But therein lies a problem, because many of these genes have relatively poor mouse homologs. So this is a heat map here showing the homology at the protein level between human and mouse Alzheimer's risk genes. Now, these genes that are dark green, we're in pretty good shape. The mouse version of those genes is very, very similar. And so likely studying traditional mouse models with manipulating those genes is going to be highly predictive of what might be occurring in Alzheimer's patients. But as you get into these bluer colors and even these gray colors, these are genes that have very poor homology. In fact, the gray arguably don't have a homologous mouse gene. And the blue are very, very low, on the order of sort of 60 to 50% homology. And so that has led some in the field to wonder, well, for some of these genes with poor homology, are we going to need to turn to different models that better represent the human version of these genes and proteins? With that in mind, we and many other groups around the world started to think about whether stem cells might be able to be used to study these genes in human microglia. And so, of course, you're probably all very familiar with the concept that one can take a patient cell, such as a skin cell or a blood cell, and by applying some specific transcription factors, which we often refer to as Yamanaka factors after Shinya Yamanaka, who developed this approach, you can get these cells to sort of reset their clock, turn back to a pluripotent state, a state that's essentially representing the stem cells in the early blastocysts. Those cells, being pluripotent, are capable of giving rise to any cell type in our body. That was established now, I think, almost 14, 15 years ago. But what we started asking just about 7 years ago was can we take those pluripotent stem cells and make them into microglia? Can we figure out how to provide the right cues in a dish to drive them into patient-derived microglia so we could start to study these Alzheimer's risk genes in human microglia? So this slide sums up a lot of work from a very talented graduate student in my lab, Edsel Aboud, who spent essentially most of his thesis trying to figure out how to drive iPS cells into microglia. And what Edsel did was to look at the literature and what we know about where microglia come from during mouse development. And there's actually some beautiful lineage tracing studies showing that microglia originate as primitive hemopoietic progenitors within the yolk sac. Those then differentiate into erythroid myeloid progenitors that infiltrate the central nervous system, and then the blood-brain barrier forms, trapping them in the central nervous system, and cues within the brain then help these cells to further mature into what we know as microglia. And so what Edsel did was to develop a protocol that mimicked this, albeit in a dish, essentially creating a two-step protocol that first generated these primitive hemopoietic progenitors and then moved them into different media that mimicked the brain environment, providing some of the key growth factors that had been implicated in microglial development. Now, as often the case in science, if you think you're onto something kind of interesting, chances are many other groups are doing similar stuff. And sure enough, over the course of a couple of years, many groups published their own version of how to make these microglia. And there's of course pros and cons to the different protocols, but all of them essentially try to mimic this developmental ontogeny and make a microglia-like cell in a dish. So we had to, having established a way to make these microglia, we had to decide, well, which is the Alzheimer's gene we're first going to look at. And so I should say we kind of picked the easy path in that we went after a gene that has a pretty dramatic effect on Alzheimer's risk. So the gene TREM2, which stands for triggering receptor express on myeloid cells 2, mutations in this gene had been shown to increase Alzheimer's risk by about threefold. So they're a pretty dramatic mutation, although they're relatively rare. Only about 1 in a couple hundred Alzheimer's patients have these mutations. But groups had begun to study TREM2 and learned quite a lot about how TREM2 functions and how it might influence Alzheimer's disease. It's a membrane-tethered protein that will recognize specific ligands, and some of those ligands are shown here. It's a lipid sensor, so it can sense different lipids. It can interact with apolipoprotein E, which is another well-known Alzheimer's risk gene. It's been shown to interact with A-beta. Here it says oligomers, but also other forms, fibrils of A-beta, and also some evidence it might sense double-stranded DNA. And upon detection of this, will signal downstream through the signaling cascade via this DAP12 adapter to lead to phosphorylation of spleen tyrosine kinase, or SYK, and that leads to several downstream pathways, including PLC gamma, IP3, which then leads to calcium release. So what we wondered was, can we start to study TREM2 using these iPS cells? Now, the mutations that have been identified occur within the ligand binding domain, and quite a lot of evidence has suggested that they are partial loss of function mutations. So we began our studies by asking what happens if we knock out TREM2 in human microglia. So at this point, we turned back to some control iPS lines, 3 different lines, and we used CRISPR gene editing to make homozygous deletions of TREM2. And you can see here just a Western blot validating that, yes, we have knocked out TREM2 at the protein level in these 3 different iPS lines. So our next step was to see, well, by knocking out TREM2, have we impaired this downstream signaling pathway? And so we looked at phosphorylation of SYK, which is the most immediate response of stimulation. And in order to stimulate TREM2 with high specificity, we used an antibody to TREM2. It's a polyclonal antibody that actually causes the receptor to cluster and can drive SYK phosphorylation. And you can see that here in the Western blot, that we have TREM2 wild type cells, and within just 5 and 15 minutes of exposure to this antibody, we have a nice phosphorylation of SYK. In contrast, in the TREM2 knockout cells, there's no induction beyond the 0 time point in the baseline. So we've lost that induction of SYK in response to that antibody. Now, this eventually leads to downstream release of calcium from the endoplasmic reticulum. So we also did a similar study looking at calcium release in response to this antibody stimulation. And you can see here in control cells that, yes, upon TREM2 antibody treatment, you get this calcium release from the endoplasmic reticulum using a calcium reporter. So what does this do to the function of human microglia? Well, we started with RNA sequencing studies to try and ask, well, how are the genes changed 24 hours after the stimulation? And we saw a number of different gene expression changes, but this is a subset of that list, looking at what we call differentially changed genes that are changed both with the knockout, but also with the stimulation. So what we've organized here is we've got several groups here. Let me just unpackage this. We have in black, all of these ones above the black bar are TREM2 wild type cells. All the ones above the gray bar are TREM2 knockout cells. And then we have either not treated them in red, or we've treated them with a control IgG antibody in yellow, or we've treated them with the TREM2 stimulating antibody in green. And we've done that for both the knockout and the wild type. And so here we've organized the genes to show which genes change in the wild type with the antibody stimulation, but have an opposite state change in the knockout, suggesting that, yes, TREM2 is really regulating these genes. And we figured that those genes are going to be some of the most important ones to study. So you can clearly see in blue here on the right are genes that are downregulated upon antibody stimulation, but are upregulated in the knockouts. And in yellow and green are genes that are upregulated with stimulation and downregulated in the knockouts. So, we get a whole big list of genes. And of course, one of the easiest things to do is to then look at the gene ontology. What are the pathways being implicated by these sets of genes? And so when we do that, we find that there are several interesting pathways starting to show up. So chemotaxis, with movement of these microglia to a given stimuli, evidence for altered interleukin signaling, so a pro-inflammatory cytokine, evidence for altered ERK phosphorylation and MAP kinase pathways, suggesting potentially some of the cell survival pathways and growth factor pathways, and some other pathways implicating phagocytosis. So based on this, we then decided to go back and look at some functional assays to see, do these gene expression changes lead to functional differences in the TREM2 knockout cells? And so we used a standard approach to measure phagocytosis, the ability of these microglia to internalize and degrade various substrates. I mentioned that TREM2 recognizes ApoE. So we teamed up with some colleagues that had some fluorescently labeled ApoE. And sure enough, we could see that in the wild type cells, there was less phagocytosis, less internalization of ApoE, almost none in the knockout versus the wild type. We also turned to amyloid, of course, being interested in Alzheimer's disease, and find that the knockout cells internalize less fibrillar amyloid, just shown in green here. And then actually in collaboration with the left, we were able to look at human brain-derived synaptosomes. Remember, I'd mentioned that microglia can degrade synapses and help in synaptic pruning. And it turns out that had the biggest effect, that the knockout cells had a real impairment in the ability to phagocytose and internalize synapses. So several different substrates, we see impaired phagocytosis in these TREM2 knockouts, suggesting this could be one of the functions that's underlying TREM2's effects in Alzheimer's disease. Now, the other thing we'd seen was some ontology implicating chemotaxis. So for this, we turned to, we worked with a collaborator, Hang-Sang Cho, and his postdoc, Yu-Jung Kang at UNC Charlotte, who had developed a really nice microfluidic system to study migration of cells. So they've essentially created the system where you can put one set of cells in the middle of this microfluidic system, and then there are little channels connecting to an outer well where you can now add your microglia. So what we had done was to add either amyloid itself or cells that overproduce amyloid, human cells that were engineered to overproduce amyloid, into this middle chamber. And then we can add our microglia into this outer chamber and simply ask how many of these cells move to the middle chamber over the period of about 24 hours. And so what we found was a pretty dramatic difference between the wild type cells and the TREM2 knockout cells. So here, in response to Alzheimer's neurons that are overproducing amyloid, we see early on, it takes a little while for these cells to ramp up the amyloid. And so there's a little bit of a difference, but by 9 weeks in culture, where these neurons have been growing for 9 weeks, and now we've added the microglia for just 24 hours, those microglia have been strongly recruited in the wild type case, but show very little recruitment in the knockout case. So TREM2 has impaired their ability to detect these amyloid producing neurons, and what we're not clear on is that amyloid, is that other signals from these neurons that are recruiting that at this point. So we started wondering, well, what is mediating this chemotaxis? So what are some of the receptors or ligands that might be involved in this? And our RNA sequencing data had pointed us towards one potential candidate, which is CXCR4. This is a chemokine receptor that's very well known to be involved in chemotaxis of various cell types, various immune cell types. And Amanda, the student working on this, confirmed that CXCR4 levels were decreased in the TREM2 knockouts, both by histology, but perhaps more importantly, by flow cytometry. When you look at the levels that are on the membrane, we have about half the levels of CXCR4 on the TREM2 knockouts. And so the next step was to ask, does that mean these cells are less responsive to the ligand, which is called SDF1 alpha? And so teaming up with Mike Cahalan's lab at UCI and Amit Jaraman, his postdoc, we exposed our cells to this ligand and measured calcium responses, because this ligand receptor interaction also leads to calcium release. And what you can hopefully see here is that there's a nice strong calcium release very rapidly in the wild type cells upon SDF1 alpha exposure, but that is highly muted in the CHEM2 knockout cells. So they're not responding as much, likely because they have about half as many levels of the receptor on their membrane. So we thought this might be playing a role in the migration deficit. And so one of the ways we followed up on this was to turn back to this microfluidic system and ask what happens when we have wild type cells and we inhibit this CXCR4 signaling pathway. And so there's a nice clean antagonist to this receptor, AMD 3100, and we exposed wild type cells to this AMD wild type microglia. And what we found is that that caused a deficit in the migration. They were no longer being recruited at the same rate to the Alzheimer's cultures. Now, the levels didn't come all the way down to where they were at baseline for the TREM2 knockout, suggesting there may be other chemokines and receptors involved in this. And sure enough, we've had some follow-up work that's currently in review looking at another receptor ligand pair, which is P2RY12 and ADP, that also seems to be disrupted in these cells. But certainly this data suggests that CXCR4 signaling is important for the chemotactic deficit we see in TREM2 knockouts. So everything I've shown you has been these iPS cells in culture, and they can be very straightforward to do some high throughput experiments, but ultimately in vitro microglia are not the same as in vivo microglia. And this was probably most clearly shown in this study from Chris Glass's lab, who took human microglia from the brain and then cultured them for different periods of time and examined RNA expression in these cells. And what you can clearly see is the ex vivo samples, which have been isolated from the brain, very quickly mRNA is isolated, versus cultured cells, even for just 6 hours or 24 hours, the gene expression begins to change very rapidly. And that makes sense. We know microglia are very plastic cells, very responsive to their environment. And sure enough, an in vivo microglia seems to respond quite differently and change once they're put in culture. So that got us wondering, is there a way to circumvent this? Is there a way to deal with this problem and provide a little more physiological way to study human microglia? And so we specifically wondered, could we mature some of these microglial precursors, the human hematopoietic progenitors, within a mouse brain, and would that promote a more ex vivo-like brain signature, so more like the human brain signature? So we went back to our protocol and said, well, what happens if we take this intermediate step that's sort of similar to the cells that start to infiltrate the brain and put them into a very young mouse pup while those mice still have microglia developing? Now, of course, we need to use a specialized mouse because we're putting human cells into the mouse. So the mouse needs to be immune deficient, which is what a couple of these knockout genes enable. But they also have to express this human CSF1. This is a critical growth factor for microglia. And it turns out that the mouse version of this ligand will not activate the human receptor. So we had to use this humanized version in order to get the human cells to survive. So we did these transplants, came back a couple months later to look at the adult mice and see what was going on. And I was really very pleasantly surprised by the degree of chimerism we were able to achieve. So this is a sagittal section of one of those mice, and the green is showing a human-specific antibody to P2RY12, which is a homeostatic microglial marker. And you can see that we've got pretty widespread distribution of human microglia throughout the forebrain, the olfactory bulb, etc. You'll note that there's not a lot of human microglia in the hindbrain and the midbrain. And we think that's because there's competition for the mouse microglia and where they get to first. So we injected in the forebrain, those cells are able to migrate and set up sort of a niche before the mouse microglia fill the niche. But by the time they reach the hindbrain and midbrain, mouse microglia, endogenous mouse microglia have proliferated and taken over the niche. We were okay with this because, of course, Alzheimer's is really focused on the forebrain. And so that's what we're intending to study here. So we were quite happy with the engraftment we achieved in the forebrain. If we quantify this and look at some of the different markers, we can confirm that, yes, these cells express typical homeostatic markers of microglia. They express the myeloid transcription factor, PU.1. We can use GFP-labeled iPS cells to make it very easy to identify the human cells. And when we quantify the degree of chimerism across multiple mice, we see that we can achieve about 80% chimerism in those 4 brain regions across multiple mice. So one of the things we wanted to know is, well, these cells look like human microglia. Are they actually acting like microglia? Do they function like microglia? And so for that, we teamed up with a colleague at UCI, Sunil Gandhi, and his graduate student, Dario Figueroa, who are very good at doing multiphoton imaging. So they can put a cranial window into the skull and then perform multiphoton live imaging of a given cell. And so we took our mice that were transplanted with GFP human microglia, and they did some multiphoton imaging. And what you can see here is that the cell bodies are fairly stable, but there's some little processes kind of moving around, surveilling the local area. And that's typical homeostatic function for microglia. In fact, if we quantify this rate, it's nearly identical to the rate of extension and retraction that one sees with mouse microglia. So it looks like they're undergoing homeostatic surveillance. But then we took the next step, which was to ask what happens if we now provide a very local insult. And so there's a classic paradigm in the microglial field where one can shine a very strong laser, intense laser beam on a small focus, and then watch how those microglia respond. And so what we find when we do that is that there's this very rapid influx of microglial processes to that injured area. The rate of that influx is, again, very similar to what one sees if you do a similar lesion in mouse microglia. So what this is telling us is that, yes, the human cells are functioning. They're functioning at least in the same way a mouse microglia will. We obviously don't know the entire kinetics of this in human brains because no humans are wanting to volunteer for these kind of imaging studies. But it does suggest that these cells are acting like normal, typical microglia that can respond to local injuries. Now, if you remember, one of the reasons for doing this transplant was to try and see, will this promote this ex vivo signature, more like human microglia from the brain? And so we isolated the cells after transplantation and performed RNA sequencing. And we compared our data set to Chris Glass's data set. And so this is a principal component analysis looking at all the gene expression of these different cell types. And so let me just explain a little bit of what we have here. Over on the bottom right in red, we have blood monocytes, human blood-derived monocytes. And these are used as a contrast because monocytes are, if you like, a cousin of microglia. They're also a monocyte lineage like microglia, the phagocyte, but they clearly are quite different in terms of their gene expression. Up at the top, we have cultured human brain-derived microglia. So these are samples from Chris Glass's 7-day samples and some equivalent samples that we generated in my lab. And then in yellow, we have our cultured iPS microglia. And what we're trying to achieve are these green samples. These green samples are the ex vivo samples from Chris Glass, and again, a few similar samples that we derived. And what is most fascinating here is these purple samples. These are the human microglia that are taken out of the xenotransplanted mice. So we call them XMGs. So we've transplanted the mice, waited 2 months, pulled them back out, and isolated RNA. And you can see that this transcriptome now overlaps very nicely with the ex vivo signature. So it looks quite promising. Now, those of you who work with RNA sequencing and are familiar with these PCAs know that these are very much influenced by contrast. So because we have these monocytes over here, it kind of pushes all the microglia to one side and makes them look more similar. So we redid the analysis excluding the monocytes so that we can get a better spread of our different microglia to see how similar or different they are. And that reveals some more information. It shows, for example, that the cultured human microglia from the brain are a little bit different between Chris Glass's lab and my lab, just sort of some technical differences. It shows that the iPS microglia are a little bit different on their own as well. But what's exciting is it still shows consistently that those transplanted microglia are very similar to those ex vivo human microglia. So that's really encouraging that we have established a system where the human transplanted microglia are able to take on a transcriptomic signature that looks very, very similar to human microglia that one would isolate straight out of a human brain. So now that we've established this model, what can we do with it? Well, of course, being interested in Alzheimer's disease, we want to know how these microglia might respond to different pathologies. And we're particularly inspired by this important study that came out from Ido Amit's lab, where he identified a subset of microglia that he referred to as DAMs, or disease-associated microglia. And so DAMs are a transcriptomic signature that changes in microglia when they are adjacent to plaques. So here you see a plaque in white with some various different markers for microglia. And there are specific genes that get highly upregulated in these plaque-associated microglia, and some other genes that get downregulated. And Ido Amit's group described these genes and coined the term DAMs to suggest these are responding to amyloid plaques when they have this specific gene expression changes. So we simply wanted to know, will human cells show a similar type of response? Will they show a DAM response in our chimeric model? So we adapted our approach in order to study this. And we had a couple options. We could either cross our immune deficient mice with a traditional amyloid model, such as the 5XFAD, or we could take our immune deficient mice and inject amyloid oligomers. And there's, of course, pros and cons to those. Those of you who work with mice can probably sympathize that to do this cross and restore homozygosity for these genes took about 2 years of breeding, a lot of PCR. But the alternative had a problem in that although we could do it quicker, injection of amyloid doesn't cause plaques, and the DAM response is a plaque response. So we really wanted to mimic a real plaque environment. So we chose to go with this longer approach of breeding the mice together. So we bred the mice together, we transplanted some of our GFP human cells, and came back about 10 months later to see what had happened. And what we find is that we have very nice engraftment of our human cells shown in green here. And we see amyloid plaques shown in white here. And we can see that these microglia are starting to migrate around the plaques. You're seeing these brighter clusters of microglia near the plaques. And they show some very specific changes in protein expression. So this marker here, CD9, which is a tetraspanin, it's an exosome marker actually, is highly upregulated in the microglia, but only when they are adjacent to the plaques. You can see there's a real increase in that signature in these clustered microglia next to the plaques. And you can see that a little clearer in this high power view where the cells adjacent to the plaques upregulate CD9. And that is one of those DAM markers that Ido Amit had shown. So it suggested, yes, our cells are adopting a DAM-like phenotype. Indeed, we looked at some of the other markers that Ido Amit had implicated, CD11C, MERTK, and even TREM2 itself. And all of those are upregulated in the microglia adjacent to the plaques. So we then wondered, is it going to be exactly the same as the mouse model? You know, were we wrong to think that we need to study human cells? Can we learn everything we need to from a traditional mouse model? So we took an unbiased approach, which was to isolate the human microglia and perform single cell sequencing from these mice. And what we find is that we can detect at least 4 different subsets of microglia. We have a sort of a canonical, more homeostatic state. We have some that really upregulate MHC2 molecules. We have some that upregulate interferon. And then we have cells that upregulate DAM markers. And you can clearly see there's a lot more of these DAMs. They're shown in pink here in the 5X, the Alzheimer's model, than the wild-type model. We see a few of them in the wild-type model, but not nearly as many. And so that suggests, yes, there is a DAM signature occurring in these human cells. That's quantified here. So we see at least a 4-fold induction of the DAM signature in the presence of amyloid pathology. But so then we asked, well, what are all the genes in this DAM signature and how similar are they to the mouse DAMs? And we were actually quite surprised that the overlap is fairly weak. We have about 9 to 10% total overlap between DAMs that were identified with this chimeric model and DAMs that were identified by Ido Amit. There are some key genes that do overlap, such as CD9 that I showed, such as TREM2 and ApoE, and that's very important. But there are also some genes that are up in the mouse model, but not in the human, or up in the human model and not in the mouse. Now, there could be a lot of explanations from this. Some of this is likely species differences. Some of this is also going to be technical differences. You can see that the green circles are bigger, and that's because we sequenced more cells more deeply. And so we have more statistical power than the earlier study. And so that's why we have more genes. But overall, we were encouraged because there was a further enrichment of Alzheimer's disease risk genes in the human DAM signature. We went on to try and validate some of these by using immunohistochemistry, both in our mouse model and then also in a human Alzheimer's disease cases, and indeed could find that these markers were upregulated in the microglia adjacent to plaques in human cases. So I began by talking about TREM2 and how TREM2 might be important for Alzheimer's and how it can influence microglia in vitro. So we obviously then went back to ask, can we use this chimeric model to study TREM2 in vivo? And so we went back to our iPS cells and we did a new set of transplants with mice with both the wild type cells and the isogenic knockout cells. And what we found was quite striking. If we just look at the distribution of these cells, we see that in green, the wild type microglia at the top are, as we'd expect, gathering around plaques, responding to plaque pathology, but the knockout microglia are sort of not even recognizing that there's pathology. They're just randomly distributed, not responding and migrating around the amyloid plaques in the same way. That suggests a couple of things. One is they're unable to sense the amyloid. The other is that they might have a problem migrating to the amyloid. And as I showed you earlier, our in vitro data suggests there is a migration problem and potentially a sensing problem as evidenced by the phagocytosis studies. So, as you might expect, if the cells don't migrate to the plaques, well, then you don't get a DAM signature. And so sure enough, there are a greatly decreased number of DAM microglia in the knockout, the mice that received knockout TREM2 cells. Here we combined in the same mice, both knockout and wild type microglia, where we labeled one in green and one in red. And we, of course, did the reverse to make sure there was no issue there. But what was very consistent here was even with the co-transplant, we see that the wild type cells migrate and cluster around the plaques, but the knockout cells do not. And then the wild type cells upregulate some of these DAM genes, such as HLA, DRB1, and CD9, and the knockout cells do not. We've subsequently gone and looked at now a specific patient mutation. So the R47H coding mutation is one of the mutations that actually occurs in Alzheimer's disease, and it essentially phenocopies the results, that there's an impairment in the proximity of the microglia to the plaques and a decrease in those DAM genes. So I just want to end in the last few minutes by talking about what we're doing next, which is to recognize that everything I've shown you is really focused on amyloid pathology, but of course there's neurofibrillary tangle pathology in Alzheimer's disease, and microglia are known to influence this pathology. And so we've been very interested in trying to understand how human microglia might interact with tangles. So we've developed a version of this mouse that uses a tau mutation to drive tau pathology, and we've begun to do transplants to look at how our microglia might interact with these tangles. Now, we've also then combined both models with amyloid and tau, so we better mimic the real human disease that has both pathologies. And so here you can see some examples of our GFP microglia, some tau pathology, some amyloid pathology, and putting them all together. And we're finding some pretty interesting features, such as in the hippocampus, there's this very unique distribution of microglia where they have become elongated, and they seem to really interact with these tau-laden dendrites. So we're trying to understand exactly what's going on there. We can, of course, go and use things like RNA sequencing to examine those. And so in a study that's ongoing by Morgan Coburn in my lab, a graduate student, we have found that there's sort of a heightened response in the combined model. So once you add amyloid and tau together, you get even more DAMs, you get even more interferon cells, and you get a pretty dramatic increase in the production of IL-1 beta and a subset of cells that are producing high IL-1 beta. And so it suggests that the response to microglia to combined pathologies is by far the greatest. Although amyloid drives a lot of it, you can see it certainly with the DAM response. Tau on its own doesn't drive a DAM response, and that's informative. But when you put the two together, you get a heightened response. So the last thing I want to mention is that we're always concerned about the mouse microglia and how they might interact. Remember, we can achieve about 80% chimerism, but that means 20% of the forebrain neurons are mouse microglia. And so they might compensate in terms of responding to plaques and things like that when we have a dysfunctional human microglia like the TREM2 knockouts. And so we've now established a way to achieve 100% chimerism. So we took our mouse model and we've crossed it to this newly developed mouse model called the fire mice. And these have a very specific deletion in CSF1R that allows them to develop without any microglia. And they're actually quite surprisingly viable mice. They do have some pathology that occurs later in life, but these mice then have a blank slate. They have no microglia. We cross them in together, and then when we transplant our microglia, we can achieve 100% chimerism. So this is a half-brain confocal stitch showing human microglia with GFP here, showing a very typical distribution tiling pattern microglia, and there are no microglia, no mouse microglia in these mice. So with that, I just want to summarize by sort of going over the main points I tried to show you. We and many others have developed these protocols to produce large numbers of human microglia from iPS cells. And by combining that with things like CRISPR, we can manipulate these genes and study how these Alzheimer's genes might influence microglia in vitro. By maturing these cells in chimeric mice brains, we can also start to look at how these genetic changes might influence the function of microglia in vivo in this sort of combined chimeric system. I then showed you that the human dam signature only partially overlaps with the marine dam signature, and I think emphasizes some of the utility of these models, and actually was somewhat predictive of some subsequent studies coming out showing that when people do single nuclear sequencing of human brains, they also don't match very well with the mouse dam signature. So, and in fact, we're finding some evidence that they do match more closely with our chimeric dam signature. And then we started to dig into the mechanisms here, and we believe that the CXCR4 signaling is at least part of the dysfunction that's occurring in TREM2 mediated migration deficits, although, as I mentioned, there are likely other things that are influencing TREM2 function in important ways. And then lastly, that we are now trying to develop some additional models to sort of better recapitulate the whole combination of amyloid and tau pathology that actually occurs in patient brains. So with that, I want to, again, thank Lef for hosting me and inviting me. And then, of course, thank the fantastic graduate students that performed all this work. Johnny, Morgan, and Amanda especially were really important in this work. I mentioned that Lef and his former advisor, Carl, collaborated with us on some of the synaptosome work and that went into that TREM2 study. We have some great collaborators elsewhere, UNC, McGill, and University of California, San Diego, as well as Edinburgh. And of course, funding from NIH and the Cure Alzheimer's Foundation. So I hope there's some time, and I'd be delighted to answer some questions.
Thanks so much, Matt. It was a wonderful talk. I know the work, but I didn't know some of the last slides about your recent work and improvements of your chimera. It's amazing. It's very sophisticated models to study the human role, and it's a clean data to show the difference between there are a strong difference between the mouse microglia and the human. So now we will have the section of QA, the QA. Okay. We already have some questions here, and I will read. So Dr. Mauricio Diaz said, excellent seminar, thank you. Yeah, and the question is, can this special type of microglia explain the different susceptibility of brain regions to AD-associated damage?
Yeah, that's a fantastic question. So there's a growing understanding that there are regional differences in microglia. And there was a beautiful study just a few years ago showing that just in a normal wild-type mouse, if you isolate microglia from different brain regions, they have different transcriptomic signatures, and that likely translates to different functions. We have seen evidence for that in the chimeric model. So for example, cells that are in the white matter have a different morphology, and we're starting to see evidence that they have different gene expression than cells in the gray matter. And I think it will continue to go beyond that. We're actually using that newest model where we get ideal chimerism to be able to see what happens to the microglia in the cerebellum versus the spinal cord versus the forebrain. So yes, the environment, the brain environment affects the gene expression of microglia very exquisitely. We even, for example, see that when we transplant our cells, if they happen to be in the meninges, they adopt the signature of a meningeal macrophage, not a microglia. If they happen to be adjacent to a blood vessel, they adopt the signature of a blood vessel macrophage instead of microglia. So they're very responsive to their environment and change their gene expression. And I think, yes, that in combination with one's genetic backgrounds is going to make some changes. And I think there's a lot of interest now in what are called BAMs, border-associated macrophages, which are related to microglia, but play a very important function in the blood-brain barrier and are increasingly being implicated in Alzheimer's disease. And so I think as we do more of this work, and others in the field do more of this work, we're going to find that, yes, these other regional differences may be very important and, maybe contributing to the regional susceptibility of certain areas in Alzheimer's disease or other diseases. Where a certain gene might make you susceptible to Alzheimer's, other genes might make you susceptible to frontotemporal dementia and that regional distribution. So, we don't have a good answer yet, but the bottom line is I think there will be a lot coming out over the next 5-6 years showing that there is an important implication of regional differences in microglial function.
Thanks. We have another question from Dr. Gina Quirarte, and she said, congratulations for your beautiful work. And the question is, does this microglia also involve in other brain structures related with the previous question? But in addition, she wants to know that if you can comment a little bit more about the microglia in the amygdala, and if this type of disease-associated microglia also plays a role in other neurological disorders.
Yeah, also a fantastic question. I mean, so absolutely, microglia are playing some function in the amygdala. I would say it's quite understudied right now. Obviously, the amygdala is important in fear behavior and emotional behavior. And I think could be a lot of work that could be done on that. And I fully expect that they're going to be important in those behaviors. Their role in just simple synaptic plasticity could influence the impact of that. I should say we are seeing a little bit of hints of changes in elevated plus maze, which is a measure of anxiety. That I can't remember if that completely involves the amygdala, but I wouldn't be surprised if it does a little bit. So there's, as we start to do more behavioral analysis, I think we'll be able to get a better understanding of how different cells and different mutations might influence related behaviors. In terms of other neurological diseases, I think, yes, absolutely. Microglia are important in a number of neurological diseases. And what we've used to guide ourselves is the genetics. So we can look at the diseases and what's known about the genes and start with the very simple question of what cell in the brain expresses the highest levels of those genes. And so one good example is frontotemporal dementia. There are genetic causes of frontotemporal dementia, and two of the most prevalent genes are progranulin and C9orf72. Both of which are most highly expressed in microglia. And as people have made traditional models of these, they're finding lots of changes in microglial function. So I think one could adapt this kind of approach to study frontotemporal dementia. Likewise, we're starting to look at lysosomal storage diseases. Turns out a lot of those have very important microglial gene signature. And even Parkinson's. Some of the genes implicated in Parkinson's, LRRK2, and GBA are both highly expressed in microglia, and some very recent studies have shown that even in IPS models, that LRRK2 mutations can influence microglial function. So yeah, I think we're just starting to get a glimpse of how microglia might be involved in various neurological diseases.
Thank you. There is a very specific question for Dr. Carmen Aceves. Thank you very much for your excellent talk. Could you tell us a little bit more about the lipids that activate the TREM2 receptors?
Yeah, that's a great question, and unfortunately very difficult to study. There's not enough good expertise in lipid biology that's working on microglia yet, and I certainly don't have the expertise there. But we do know ApoE, apolipoprotein, can bind to TREM2, and then there's various lipids that have been implicated, sphingomyelins among others, but it's very difficult to study because a lot of those lipids might respond to other receptors as well, and so I think that's where these knockout approaches are going to be helpful to help us clarify which lipids are important for TREM2 stimulation, but aren't also stimulating other pathways. And I would say that we're just starting to do that. The way we're trying to do it is sort of the opposite way around through some collaborations to look at the lipid accumulation within the cells. So there's some evidence that TREM2 knockouts and mutants will accumulate more lipids. And certainly the DAM signature involves a lot of lipid responsive genes. So lipid metabolism seems to be very central to what's going on with the DAM signature. And in fact, we recently found that there's an overlap between the DAM signature and arteriosclerotic foam cells, which are a macrophage that is accumulating lipids. So I think the lipids is going to be very important. So we're taking the opposite approach of isolating our cells and then performing lipidomics to see what kind of lipids accumulate in the context of Alzheimer's pathology versus controls, or with TREM2 knockout versus controls. And so I think that will start to give us more clues. So I don't really answer the question about what lipids TREM2 senses, because I don't think we know the answer to that fully yet.
Fernando Talavera says, very interesting seminar. Thank you very much. I wanted to know if it is possible to create different types of chimeric mice to study other kind of diseases.
Yeah, absolutely. The challenge is the breeding. You know, I showed that figure of 2 years about, because what we figured out is you need 3 genes that need to be restored to homozygosity: the RAG2 knockout, the IL-2 receptor gamma knockout, and the humanized CSF1. If you have those 3 genes, then the human cells can survive. But so then when you start with that mouse, you then have to cross it to whatever your other model is, right? We've now done that with a progranulin model, you know, so we've got a frontotemporal model we're starting to work on. We're doing it on a lysosomal storage disease model as well, but it takes quite a bit of time to do that crossing, unfortunately. If one has a lesion model, say something like traumatic brain injury, then yes, you can buy the line that is available from JAX. It's called the HCSF mouse. You can buy that, breed that, and do your TBI. Then that's fine. But if you are requiring a genetic model like an Alzheimer's or a frontotemporal model, you're going to have to do that crossing. So that, I think, slows us all down and is challenging. But certainly for injury models or other things, Cooper's-own to look at white matter injury, you would be able to buy those mice from Jackson and do those studies fairly quickly.
Thank you. Dr. Juan Riesgo, the question is, what about the difference between neurons in the immediate environment of microglia?
Yeah, the interaction with neurons is a really important and interesting question, and we're only just starting to touch on that. So we're doing that a couple ways. One is that we're turning back to co-culture. So we're taking iPS neurons and putting them in the microglia. And we're already finding that the proportion of those cells matter. If you have too many microglia, super physiologic numbers of microglia, you're going to have over pruning of those neurons. So there's an immediate interaction there. And we're actually looking at how that leads to altered action potentials of the neurons. But then we're also, a new graduate student is actually gearing up to do a study I'm really excited about, in which she's taken the chimeric mice, but then also using a virus to deliver dreads into neurons. So dread genes where you can cause a neuron to fire or to suppress that neuronal firing. And then we want to see how do the microglia respond to that. There's some evidence that they might prune more synapses in inactive neurons. Can we see that in this chimeric model? So I think those kind of experiments will start to shed more light on that. Within the immediate area in Alzheimer's, we know that there are dystrophic neurites around the plaques, and there is growing evidence both with traditional models, and we also see it with our chimeric models, that one of the functions of the microglia are to surround the plaques, wall them off, and reduce that dystrophic neurite damage. So in general, when we look at our wild type cells, we have less dystrophic neurite damage. When we look at our TREM2 knockout cells, we have more dystrophic neurite damage. So in that sense, the neurological pathology is certainly being influenced by these Alzheimer's risk genes in the microglia.
Okay. There is a very specific question about if the mTOR pathway is involved in the activation of these genes.
Yeah.
That you describe it.
So also a great question. And we've not looked a lot on this other than to say that when I look at our gene expression differences and changes as we manipulate these, there are several mTOR genes that start to pop up, and some of the gene ontology has implicated mTOR. So I think the short answer is yes, it is absolutely going to be involved. And mTOR is, of course, known to be involved in autophagy, and autophagy and phagocytosis kind of overlap in their pathways. And there's a number of studies coming out suggesting that that mTOR pathway may be important in microglial function. We've not done specific experiments to address that at this point, but they are certainly popping up, and I think will be important in, for sure, the phagocytosis response and likely many other things that of how the microglia are responding.
Thank you. Dr. Aguilar is asking is raising his hand. Could you please go ahead, Dr. Aguilar? You want to ask the question directly?
Yes, thank you very much. Congratulations for your great work. And maybe this is a not easy to answer question. I have heard proposed treatments or interventions, for example, exercise, to slow down the progress of this disease.
Yeah.
And I wonder whether your discoveries about the glia could have a path for intervention to slow down the progress.
Yeah, a fantastic question. So one of the experts on the exercise hypothesis is actually Carl Kottmann, who left, did his postdoctoral, and I also did earlier postdoctoral training with. So Carl is well known for having established that one of the key things exercise does is elevate levels of brain-derived neurotrophic factor, BDNF, which is normally reduced in Alzheimer's disease, and we know is very important for promoting synaptic plasticity, neuronal survival. And so Carl is actually the principal investigator on a clinical trial that is testing exercise in early Alzheimer's disease to see whether it can slow deficits. So yes, absolutely. I agree that the literature very strongly supports that exercise in itself can be protective. What I don't know, and I don't think there's been a lot of work on, is how exercise might influence microglial function. We do know that microglia can make BDNF, and that there's some evidence that their upregulation of BDNF is protective as well. And so whether exercise is also somehow regulating microglial BDNF, we just don't know yet. It's the kind of thing that I should go to Carl and design an experiment to test exactly that, and to see how exercise affects microglial gene expression and function. I expect it will, but I don't think there's a lot of studies yet on sorting that out.
Okay, thank you very much. Congratulations again.
Thank you.
We have another question, is that could you elaborate on the difference of in vivo versus in vitro microglia? Can cultures mimic some of these in vivo situation? I think.
Yeah, great question. I mean, so we continue to use in vitro cultures because there are things you can do in vitro that you just can't do in vivo, you know, higher throughput type of studies, really highly quantitative studies. And what we are doing is now using our RNA sequencing data to try to guide how to improve the culture. Are there other factors we could be adding to the culture that will make them more like the brain environment? What we know, and this has actually worked for some other groups, have published that when you culture microglia with neurons, you help to promote more of an in vivo signature. So in fact, if you're doing a co-culture system with neurons, or even more recently, Lorenz Studer's group added in astrocytes as well. If you have a co-culture that now better mimics the brain environment, you start to change the gene transcription signature to make them much more like the in vivo situation. So I think one can make these more complex cultures to get closer to the in vivo state. I think there's still a lot of work, and I think it will be challenging to get all the way to the in vivo state, but it really depends on the questions you're trying to ask and what's going to be the best system to ask those questions in. And I think in vitro cultures, even monocultures, are still highly useful. As with all science, you often want to take your in vitro results and try and see if there's a way to test and validate them in vivo. But I think they're highly useful, and certainly as you start to add in other brain cell types, you get them closer and closer to that in vivo situation. The other approach that a lot of people are working on are brain organoids. And there's some advantages there because you can use human neurons, human astrocytes, human organoids, and human microglia. So everything's human. And so there's a lot of progress being made in that. There's a very recent paper from Genentech showing actually a high throughput organoid system with microglia, where they could look at, and they could actually even see what looks like amyloid plaques forming and seeing the microglial response to that. So I think that's very rapidly accelerating the organoid work, and I think we'll continue to see progress there. And people are starting to look at the signatures in those organoid models, and it looks like that also makes them more like the brain microglia.
Thank you, Matt. We are running out of time, so we need to finish the streaming. But I have just a very quick question at the end, because maybe you know the work of Gary Lynch with the cognitive enhancers, alpha-kines and this idea. But it seems like the microglia is also playing an important role. Maybe you know if people are working with microglia enhancers or trainer of microglia educators.
There are several companies going after TREM2 specifically. So, and one of the ways that several are doing are stimulating antibodies. So similar to some of the stimulating antibody work I showed, they have stimulating antibodies that they are hoping to drive increased TREM2 signaling, because just as there's decreased TREM2 signaling is associated with increased AD risk, the expectation is maybe the converse. Maybe if you increase TREM2 signaling, that's going to be protective for Alzheimer's, but potentially TREM2 has been implicated in frontotemporal dementia as well, and maybe some other diseases. So there are certainly several groups trying to do that. As you know, the challenge with antibodies is the blood-brain barrier.
Right.
But you have companies like Denali who have proprietary systems to increase access to the blood-brain barrier of their antibodies that are working on some of this. And then there are some groups. There's one small company that sprung out of Amgen that is working on small molecule TREM2 stimulators. So I think TREM2 is the first target people are going out after, because we know the most about it. But yes, there's a lot of interest in trying to understand these other genes and whether they might point towards other druggable targets to stimulate microglial activity in beneficial ways.
Okay. Well, once again, Matt, we are very thankful for your participation, and we hope to see you here in vivo, in vivo soon. And well, thank you. We are closing this session and inviting for people for our next activity at noon. So we will see you again in some minutes. Matt, I will be in touch with you later, and thank you very much, Matt.
Thank you so much, Aleph. It was a pleasure, and thank you all for the fantastic questions. Really enjoyed it.
See you.